So this video is to describe the procedure for the epidemic simulation exercise in the lab manual. This epidemic simulation shows a couple of important concepts. It shows the index patient of an infection. The index patient is the first person infected. And it shows the reproductive rate of an infection, also known as the R0. The R0 is the number of people that will be infected by a sick person. So it shows you how fast an infection will spread. It is demonstrated through in this exercise through a bacterial contamination of hands. So one person in the class will get bacteria on their hands, and then everyone will shake hands, and we'll see how far that infection spreads. Of course, we don't know who it is that has the infection until after we see the data. So the setup of this is each student receives a plate. You'll draw a line down the center of your plate. Label one half of the plate gets labeled one half of the plate gets labeled before, and the other half gets labeled after. And what the before and after mean is handshaking. So the first thing that you do is glove your hands, and you take a swab and you take a sample of all of the bacteria that are on your glove as soon as you put it on, before you've touched hands with anybody else. And then you take that swab and you color a film. You make a lawn across the before. Okay, and then you will go and shake hands with your classmates. So this is not a random handshaking. Each person in the class will be assigned a number. So say our little example plate here, they're assigned number 3. So the number 3 person will shake hands with the number 2 person, and then they will turn around and shake hands with the number 4 person. So you receive a shake and you give a shake. The bacterial infection comes into play because through what you touch. So sitting on the counter of the classroom are petri dishes, most of them full of colored water. One of them is full of Serratia marcescens bacteria, which turns reddish brown color when incubated. So you will receive your handshake and then dip your hand in your petri dish, which most of the time will just be water, and then you'll turn around and give a handshake. So for most of you, it will just be water. You'll have colored water on your gloves that you're passing around. But at some point in time, when the student dips his hand into the petri dish, it will get bacteria on his glove. And then when he turns around to give his handshake, that bacteria will transfer. And then when that person will then dip the hand in their colored water, but when they turn around to shake hands, the bacteria will still be on the glove and will transfer. And we'll see how many times the receiving and giving of handshakes transmits the bacteria. So everybody will have a plate that you swab your clean glove and make a lawn of before, just so that we can see what bacteria were there before you touched anybody else's hands. And then you'll receive your handshake, dip your hand, your gloved hand, into your container, which is hopefully just full of water, and give a handshake. And then after that procedure, you'll come back and get a clean swab. Swab your glove that is now covered in whatever it received from the handshake plus whatever was in the petri dish. And you will color that as your after. So for most of you, there will be, a few random bacteria on the before and a few random bacteria on the after. No big deal, no surprise. But for whoever of you was the one who dipped their hand in the bacteria, the petri dish that your numbered petri dish was full of bacteria, instead of just having a few random bacteria on your plate, you will have suddenly tons and tons and tons of Serratia marcescens, red bacteria, all over yours. And then when you give your handshake, the next person will probably have some red Serratia marcescens bacteria on their after, and then the next person and the next person, and we'll see how far it goes. So that is the setup of this. It's pretty straightforward. The only kind of surprising part is whether you're receiving and dipping your hand into red water, or whether your droplets on your hand are actually bacteria-filled media that's the same color. So hopefully that helps explain the lab procedure to you.